All right. Hello, everyone. Good morning and good afternoon. Welcome to day 2 of World Seagrass Day 2026. This is a week-long webinar themed dugong conservation and habitat restoration. Today, we have Professor Dr. Muta Zakaria with us. She'll be talking about restoration, her recent restoration, which was a decade-long work in Malaysia. It's truly an honor to welcome her here. She's a scientist who has dedicated 25 years to understanding, protecting, and restoring seagrasses. And Professor Mutahara holds her PhD in seagrass ecology and biology from University of Putra, Malaysia, where she continues to lead research on taxonomy, ecology, molecular genetics, and bioprospecting of marine macrophytes. As a founder of um, Malaysia's macrophyte group, G2M, she has built an extraordinary regional research network uniting scientists from Japan, Indonesia, Thailand, and Vietnam through her prestigious GSPS Co2Co program. She's extensively worked on monitor, transplant, and rehabilitating sweetgrass meadows, translating decades of research into ecosystems that are alive, recovering, and thriving, with over 200 publications to her name. She's also a member of the World Seagrass Association and a trusted voice in seagrass conservation across Southeast Asia. Throughout this week, to reiterate, we'll learn why do we have to monitor seagrass meadows, how to monitor them effectively, how do we protect what remains, and how do we restore what has been lost. We are deeply privileged and genuinely excited to learn from her. Please join me in welcoming Professor Dr. Dahaya Zakaria. Ma'am. Hello. So I'll
Hello.
share your slide. Yes, ma'am. I'll remove myself from the stage and you can go on with your presentation.
Okay, thank you, Miss Sameha. Okay, today I will share my research on seagrass assisted recovery and monitoring for rehabilitations. I'm using a case study of the Merambong Seagrass Meadow, Sungai Pola Estuary, Malaysia. I'm Professor Muta Zakaria from University Putra, Malaysia. Okay, this is my main points today about the what is seagrass and seaweed, important of the seagrass ecosystem, challenges and risks faced by the seagrass, what happened if we lost seagrass, and of course my main topic is seagrass assisted recovery and long-term monitoring in the Sungai Pola Estuary, Johor, Malaysia. So why I use this photograph? Because of I will explain about the multi transplanting of this species. Okay, um, seagrass, as we know, is a flowering plants with a vascular system interconnecting all organs. For example, their stems, leaf, and roots, and develop well canal systems. They usually green in color, produce flowers, fruits, and seed. However, when we compare to the seaweed, or another word we can so call macroalgae, actually is multicellular macroscopic exist or stolon, rarely extensive rachis-bearing thallus or lamella, and then the color might be brown, red, green, and many more. So they never produce flower, but they have sporangiums and spore. So this is an example of the dioecious and Halus eucharides, and also the monoecious Halophila beakeri. Okay, what is important about the seagrass? In ecology, they are actually is a water filter, and then they stabilize the sea bottoms. And then in terms of the other thing is the food for the dugong and green turtle. Dugong, without seagrass, dugong cannot survive because the only food for the dugong actually is seagrass. And also they are support traditional fisheries. This is an example of the research in Sungai Pola Estuary. They have many fishes, crustaceans in that particular area. And of course, when you study seagrass, always related with the biodiversity. So in Malaysia, we have 17 seagrass species, 10 species from Hydrocharitacea, 6 species from Pseudomonas and 1 species from Rupiaceae. This one, and Halophila ovalis, the Halophila spinosa, and Halophila major, actually my species when we transplanting in the Merambong shoal. So in Malaysia, for now, we have 172 areas. Actually, we have more. And then when we look here, in Peninsular Malaysia, mostly related with intertidal, uh, lagoon, and also subtidal shoals. Okay, and then what is the challenge or seagrass ecosystem between climate change and human activities? You can see, based on the natural disaster, Malaysia always have a big flooded. So this is an example of the flooded, and then coastal developments also happen. Why this coastal development happen? Because of the flooded area. And then this is an activity, human activity in seagrass ecosystem. That's why our seagrass become decline, and then that's why we have to do the transplanting. Okay, this is an example. Seagrass in Malaysia, 2014, we had a big flood in Peninsular Malaysia. So this is the area where the Halophila ovalis grow, but now not at all. They are actually no longer in that particular area because of big flooded covered by the sand, and then the local people, local authority need to dig the area to become a relocation of the local fishing piers at the Merechang Lagoon. And then an example, another importance, if we lost the seagrass, so the biodiversity and fisheries support also will decrease. And then that's why how to make sure this one, the seagrass still survive. So we have to reduce coastal pollution, implement sustainable fishing regulations, establish marine protected areas, and then we need to conduct more and more awareness programs, and lastly, rehabilitate and restore degraded meadows. An example is the transplanting. So now my main topic is seagrass assisted recovery and long-term monitoring in the Sungai Pulau Estuary, Johor, Malaysia. For your information, in this area, actually we have two big area. One we are called Tanjung Adang, and another one we are called Merambong Shoal. This is why we start to thinking to transplant this area because of land reclamations in this particular area. So we collaborate with the industry for a city, how to save the seagrass. So this is area, the seagrass, the meadows, and then when the land reclamation happened, this seagrass divided into three. We are called, number one here is Merambong A, number B is Merambong, number two is Merambong B, and number three is Merambong C. So this is when we planted, I planted the seagrass from here, Merambong A, and also Merambong B. Let I explain to you. Okay, when in 2014, the history, the Merambong seagrass shoal happened the reclamation activities, this one, and then increase the turbidity, direct bearing of the seagrass and associated fauna. So we can see here dams of a growth of seaweeds here, and then the animals also died. They cut, for example, here in Merambong B, actually a causeway for the truck, for the lorry to take sand from the sea. So this is direct effect of the reclamation. For your information, this is my transit line because we conduct research since 1995 here, and then we really focus in 2012. So when 2014 happened, the land reclamation, all my transit here is lost. So that's why we our group called by the Department of Environment Malaysia to see, to study, to save the seagrass in that particular area. So how our strategies is, so during that time, we start to do a series of awareness program on the importance and seagrass ecosystem services. For example, we go to we join with the Tanjung Adang port, uh, because of in that particular area surrounding by the port of Tanjung Pelepas, and also went to the forest city to tell them, local people, this we need to save the seagrass in this particular area because of they are very important. And then when times come in 2016, 1015, we start to transplant the seagrass in situ. So previously, before that, we need to conduct the field observations. We study the phenology and propago collection. Then we bring to the laboratory to culture and germinate all the Enhalus eukaryotes. And then after that, we did the field transplanting, and then we monitor, continuous monitoring since 2015 to 2025. We have two phase. First phase from 2015 to 2020, we monitor monthly in this particular area. But in 2021 until 2025, because the seagrass is already okay, so we reduce from monthly to the every 2 months sampling. So why study phenology is very important? Because my subject is I want to use the Enhalus eukaryotes. And then just now I mentioned to you in first slide, Enhalus eukaryotes actually is a dioecious, meaning that male and female in different plants. So how to make sure we can save the Enhalus species when we want to do transplanting? So that's why we study the phenology. We want to know the event where the male, female, and then the flowers or mature fruit happens. So we can see here seasonal reproduction patterns and propagability actually throughout the year. So when we see this one, male, usually because of males, they have a very short peduncles. They are stay in the bottoms, and then female have a very long peduncle. Why the biological like that? Because of during the male disperse their pollens, so the female can catch. So that's why they have a long peduncle. And then when we collect the fruits or propagules, we bring to the laboratory, and then we culture, we germinate. Why we want to use the what we call the propagules? We try, actually in first phase, we try to use 3 types of culture system. For example, we use the what we call plant, the plant itself. We use seed, and also we use a seedling. So we try which one is the best. So that's why we have a study on the laboratory culture. Is it the seagrass can grow, or they can grow individually, or mix? And then in the reality, at Merambong show, actually during that particular time, we have 8 species of the seagrass in that particular. So they are mixed each others. And then when we study the phenology, we study the Enhalus eukaryotes life cycles. Why? Because we want to know where is the pollen release, fertilization, fruit, and then seed dispersal. So we use fruit to the cultivation or transplanting because of they can produce many seedling for planting materials. So when we using vegetative parts, will cause population fragmentations and reduce reproductive capability. So sustainability of planting material is very important. So that's why we study the phenology part, because we want to collect, for example, if we want to plant the individual of the Enhalus, we might be we can collect the males, and then later on the female cannot survive, they cannot reproduction happen. Why? Because of no male in that particular area. So that's why we study the phenology to collect all the fruits, and then we can germinate, because of might be in one fruit we can have many, or both male and female plants. So how we monitor? We monitor by using ground permanent transect and quadrat. Also, we use the satellite imagery, and also we use drone. So this is we have group, my group with this person, Professor Jabbar Sidik Bujang. So this person will in charge on the monitoring by using this drone, and then myself and the group will we go to the ground permanent transect and quadrat during the transplanting activity. So you can see here, when the land reclamation happened, the seagrass of Merambong shoal divided into three. Merambong A, this is a facing second link. Merambong B, the reclamation area here, and then Merambong C facing Tanjung Adang Laut. For your information, before during the activity of the uh, sand suction from here, this is a lane to bring sand to the land. So we start to planting the seagrass here. So we do the experiment here, and then in 2018, we asked the contractor to remove all this area, the causeway, and then they become similar in that particular area. So that's why we start transplanting the seagrass. So when we study, actually, first we study by using the inhalers only. Then we start to think, because of inhalers, their life cycles so long, 3 to 5 years, they can produce flowers and fruits. So that's why when we look in the environments, actually seagrass grow, multi-species grow together. So that's why we start to mix species between inhalers, we are called is stabilizer species, and then Halophila ovalis, Halophila major, or Halophila spinosa, we call as a cover species, transplanting together. For example, here, why? Because of they are very fast growth when compared to the inhalers Acoridis. This is we use a technique by using this one. So we collect from the donor site, and then also we use seed. For your information, when we use seed, 0% survival after 1 to 3 months, so no survival rate. And then when we use a seedling, actually we plant seedling of the inhalers from here, 9 area, 1, 2, 3, 4, 5, 6, 7, 8, 9, and then in between of the seagrass we transplanting the cover species of the Halophila ovalis, Halophila major, and also Halophila spinulosa. So when we study this one, actually it's not only one time. We do the transplanting activity so many times. When we collect the fruits and then we germinate in the laboratory, within 2 3 months after that, when we have a seedling, we culture, we transplanting in situ. Might be 2 months, every 2 months, every 3 months. When we have the bigger size, the juvenile size of the seedlings, so we can transplanting in situ. So we work together with the industry, we work forestry, we work together with the DOF, the DOE, Department of Environment, my student, international student, and so on. Why? To make sure the seagrass become can grow when the land activity very still happen. So this one, my manual book, how we conduct the seagrass activity together with other peoples. And then this is how the seagrass yang we call the coast, the route one, the causeway, 2.58 hectares here. So all this one during low tide, so we together with my students and local people start to planting here. We call this one is a Merambong B. And then when we collect sample, uh, we identify what type of species first here, and then what is the cover monitor, and then we planting here more or less 2 or 3 months after that they are grow, and then during next year April, from February to April to May, actually is a season for the Malay bivalve collection from the local people. So of course my seagrass during that particular time also collected by the local people. So it's okay because of this is the reality when we conduct the activity transplanting in the particular area. So when we transplanting with industry, I'm very happy during that particular time. Approximately 75 to 80% of anthallus seedlings survive in some area together with this Halophila ovalis and also Halophila major. So when we look here, we collect the seagrass still there. So we collect the Halophila ovalis with the dense area and then bring to this area. Bring to this area and then replanting again and again. And then of course, in other area in Merambong A, we also planting in the bare area to make sure the seagrass can expand and expand. So not only 1 year, we still continue transplanting the seagrass in 2022 and also in 2024. Why? Because when we see the seagrass in that particular area already lost, or because of problems, many people collected the gleaning something to collect the gastropod and bivalve. So that's why we transplanting. So this is here my colleague how they transplant the seedling of the Anhalus ochoroides. We using 1 meter time 1 meter quadrat size to transplant all the seagrass. This activity actually together with the Sarawak corporations. So not we transplants start for this one for 60 months. In this one, we can see throughout the monitoring period, seedling survival remain consistent after 8 to 9 months, with Merambong B showing slightly higher rates, 66% than MA. So MA means Merambong A, and then we can see here the percentage cover and mixed species here. We have in especially in Merambong B, in the special area. So we planted two things. We are called plot A and also plot B here. So the percentage cover of mixed species, including Anhalus ochoroides, Halophila ovalis, Halophila major, and Halophila mustata at Merambong B after 6 months still survive. And then we are very happy. I don't know, might be the other people not happy when we planting the seagrass in Merambong B. After 4 months, dugong come in. We can see the dugong trail. So meaning that we are happy because of dugong have a food to eat. So we can see here Merambong B consistently maintain higher Anhalus ochoroides cover across both early 10 to 30 months and late 30 to 60 month phases, reaching 80% in some plots. And then two representative plot at Merambong B illustrated contrasting development pattern. And then some of the area in Merambong B produce a flower. So we observe flowering after 4 years of transplanting for the Anhalus ochoroides. So here you can see into April 2002, we can see the matured fruit in transplanted area of Anhalus ochoroides. This is a male flowers. And then this is in September, Halophila ovalis cover around 80%. So that's why the study transplanting not only can study for 1 month or 1 year, but depends on the species we used. For example, if you use enhalers, meaning that we take more time. And then at the same time, to make sure the seagrass is very nice, is it okay or not, we also monitor the cover percentage of the seagrass throughout 2015 to 2024. For your example, we use the brown blanket scales, scale 1 to 5. 5 mean that very good cover with more than 75%, good cover 50 to 75%, moderate cover with 25 to 50, low cover 5 to 25, and poor cover less than 5% cover. So monthly activity, actually we conduct this one. Then in 2021-2025, we conducted every 2 months to see the seagrass in also other area near to our transplanting we did during that particular time. And then we not only monitor this one in one Merambong, but because of Merambong already divided into two, so we monitor this one in Merambong A, 3 transit lines, Merambong B, 2 transit line, and also at the Tanjung Adang Laut. They're very pristine, very not related with the Merambong land reclamation. So that's why we use this one is a control area, Tanjung Adang Laut Shoals in the figure F and also G. That's why the seagrass in this particular area always in the good, very good conditions when compared to the Merambong A. Merambong C still okay because of they are quite far from the land activities. So status. This one we are very lucky because we can see the seagrass when we transplanted produce fruits here, and then seagrass cover very good with mixed Halophila ovalis, Halophila major, or sometimes with the Thalassia hemperchai of the or Halodiol univivis in associated with the Enhalus eucharides. So in this one, you can see the green one actually is the seagrass good stand of transplants and Halus eucharides, Halophila ovalis, and also Halophila major. The spinulosa grew well in the water covered area. So this is during that time we are very happy because of our transplants areas very nice, and then we can see by using the drone, we monitor by using the drone the seagrass. Here is our transplant area, and then of course the boundary of the seagrass and Halus eucharides. So during year, mostly we can see the female and also the male release their pollens in during the low tide. And then not only that one, when we see the seagrass very nice, so the dugong trail come in here, and then we can see seahorse also in this particular area. And then this is a Venus clam. This one actually collected by the local people during February to April. So that's why from first, when I transplant Halophila ovalis, Halophila major in Merambong B, actually my seagrass also collected by the local people because of they are collect the Venus clam. And then the disease, a very famous conch shell, is very important for the local people. They are sell about 10 to 20 ringgit Malaysia per kilo. And then of course in this particular area, in 2015 we recorded more than 100 species of fauna in that particular area. For your information, we not only study on this grass transplanting or monitor, but also we study the fauna in that particular area. Other click my click, Dr. Nelina study on the impact of the reclamation and also impact of the transplanting. Is it they are more increase or not in terms of fauna? And then not just that one, in 2025 in March, we can see here the seagrass grow well, and then in the outer boundary bay and Halus ecoredes, and then here many species, a lot of in this one Halophila major, and then we also discover another species grow in this particular area. It's not 8 species, become more and more. So this is in other area, Merambong C. This one is a Merambong A. You can see here is Merambong A, facing second link, and then this is Merambong C, facing Tanjung Adang Laut. The seagrass is very huge, and then always produce flowers and fruit. So we can collect this one for our transplanting activity. And then when we study this one in 1996 to 2025, we discover a species, Halophila obiqueri, in two area, in 2015 near to the area, and also Halophila decipiens in the Merambong A in 2022, and Halophila major in 2015. And then recently we discover Halophila niponica in that particular area in 2023. And then this is how they are looks like during low tide. The Halophila biqueri, the Halophila spinulosa, Halophila decipiens, Halophila major, and also Halophila niponica. So the seagrass of Sungai Pulai, now you can see the Merambong shoals here, and the Tanjung Adang Laut, we are called Sungai Pulai. So we have 13 species now in Sungai Pulai, include Halophila inhalus ecoredes, Salacia hemprichai, Halophila minor, Halophila ovalis, Halophila spinulosa, Haledium pinifolia, Haledium uninavis, Semorosis cerulata, Seringorian acetifolium, Halophila major, the recipients, Halophila biqueri, and also Halophila niponica. So implication for the coastal rehabilitation under global change, we can see this research is emphasize the importance of long-term data sets in restoration science. Monitoring effect that span years or decades offer crucial insight into system dynamic recovery threshold and feedback mechanism. So using various planting methods like seedling and mixed species design can enhance ecological performance and adaptability in unpredictable future conditions, and successful transplanting effort in heavily impacted coastal area show recovery is possible with tailored rehabilitation and ongoing monitoring. So as we know, seagrass meadow are vital for carbon storage, shoreline protection, and biodiversity. That's why we need to protect the seagrass. Then this information is vital for improving management strategy and ecological model as habitat restoration in the region. So just for your information, in area, in other locations might be different method we can use because it depends on the biological of the species and also in the environment. So we need to protect and conserve the seagrass ecosystem because of our life depend on it for food and many other benefits. So saving seagrass means saving our seas. So that's all from me. Thank you very much all for the University Putra Malaysia Garden Pacific View and Forest TT. This is my sponsor, my collaborator, Tanjung Adang, Mosti, and so on. And of course, the organizer of the webinar. That's all from me. Thank you. Hello.
Thank you, ma'am. Okay, I have to say to take some time to digest everything. I mean, there was so much to learn from your presentation, from your study, and especially because I mean, you have an aquaculture background. I know that, and it shows when a person from aquaculture background has taken interest in seagrass restoration. Every single detail, the attention to detail is, I mean, it's admirable what you and your team have achieved. Um, I was supposed to start from my session and give a bit of a background of what's happening in the Indian context of everything. But I was, I think, a bit too eager to start with your presentation, and I'm glad I did that. So what's happening in India is that probably we are losing where there is a seagrass meadow decline or health degradation happening. And so in India, we have seagrass habitats that look drastically different from each other. So on the east coast, in the Andaman Islands, which look more like the Malaysian counterparts, we have mixed species meadows, but they are light saturated meadows, and so you'll see the halophillas colonizing, greater depths like 10 meters and maybe 15 meters too, right? Because they're all light saturated. Light is not a problem there. There is sparse meadows, but still we have mixed species meadows. Then there is again light saturated, but very densely homogeneous, very high density meadows in the South India, and they're homogeneous. I mean, you can some parts where you can see meadows extending from South India to Sri Lanka, and these are one of the most continuous and meadows within the Indian subcontinent, and even for dugongs. And then we in the northwestern, at my field study, where I also conducted the phenology study for an entire season in the northwest India, we have seagrass meadows which are not along the coastline. No meadows are with the coastline. Everything is reef top, or there are insular, islandic, or reefal meadows. So we don't have northwest India. We don't have very many deep water meadows because we have tidal constraints and very low light actually. So very high turbid waters. So we just have four species, right? So we don't get mixed species meadows. In extreme conditions, we have hypersalinity issues and low tide exposure issues. So we just have halophillas, Beckeri discipens, and ovalis, and Harah nervus, which is unlike South India and the Andamans. So you see, all these habitats are completely different from each other. South Indian meadows don't have that kind of tidal fluctuations or light restrictions. Um, so my case is that what I'm trying to say is that restoration at each habitat must look very different in the future. There cannot be one solution for all. And even regionally, if I even pick, say, suppose someone is working, and there are amazing researchers who have already started their work in restoration in South India. So there cannot be, even at regional level, one solution and one method for all kinds of habitats. Your work is testimony to that. So we understand um, that substratum matters, the texture of the substratum matters, the phenology matters. Why I brought up, I think, the northwestern meadows, the seagrass hotspot in northwestern India, where I've worked, is because your study showed that the seagrass meadows are quite persistent. They fruit. I mean, phenology is important. You need to know when the fruiting is happening, when the flowering is happening, when you can pick the flowering shoots or the fruiting shoots or the seedlings. You need to have information for that. For my case in Gulf of Kutch, they are not persistent meadows. None of the meadows are persistent. In monsoon, we lose all of them. There's seed bank throughout the summers, say February to April, and then in monsoon we lose all of them. So, and in winters again, cold water, cues are required, and light is not a problem in winters anymore. So then they germinate, Zakaria, in all kinds of habitats. I mean, I'm not just talking about intertidal. So, I mean, what will you advise? Will your advice be for I mean, this is not just my case; it's happening in a lot of places. Just for researchers who know that they have to restore certain habitats, but then we don't have persistent meadows. So we know flowering is during the, there's just a small period of flowering period, just 2-3 months, and that's it. So how do we, counter this situation
Okay,
where
thank
the, yes, reproductive phase is short?
Okay, thank you very much, Samiha. Actually, before I conduct the transplanting activity in 2015, actually
Yep.
we culture the seagrass in the laboratory first. We not culture and
Mm.
harvest this because of they are
Mm.
very long, take 5 years the life cycle. But we culture the seagrass like Halophila bicri. We culture the Halophila
Mm.
bicri in the laboratory. Why? Because of during I did this activity, I did this research since 1995 when I did my research degree. So I have a culture because of this Halophila bicra. Usually they are grow in the estuarine area. They have two types of their life cycle. One you are called annual, the other one you are called perennial. Annual meaning that uh, they mature, they produce the flowers, fruits, and the minute they sit, then they die.
Mm.
Then the seed will dormant in the substrate
Yeah,
within
in the
1 to 3 months.
yes.
So during that particular time, we cannot
Mm.
transplant. We cannot
Yep.
transplant the seagrass during
Mm.
that season. And then another one, they also have the perennial one. So the perennial one meaning
Mm.
that they mature, produce flower, produce seed, disseminate the seed, but they can grow by using their vegetative parts. So we have to
Mm.
know that one. So we study, is it they are
Mm.
they are like to stay in a salinity zero or
Mm.
hypersalinity. So we conduct activity that one we culture in the laboratory for the Halophila ovalis, Halophila decipiens, Halophila bicra, and all Halophila except Rupia maritima. So that one. So when we understand the biological part, when understand that one, so we start to thinking how to help
Mm.
the local people, how to help the seagrass because of when we have the seagrass, of course the fishes will increase. Many
Yes.
fish in that particular area. So become a benefit to the local people. So when we already understand about the life cycle of the all species because of for your information not only they in Malaysia not all area mixed species, no. So some species for example they only grow in one species, monospecies we are called Halophila bicra in Pangkalan Nangka.
Mm.
Previously one species and then after that the seedling from the seed from others area until union vis Hadilipinifia come in together. So that's why they become two species in one area, three species, or the highest one is 13 species like Merambong. So if you want to study the transplant activity, number one, we have to study the biology of the sample first. Then of course we have to study the ecology part. Is it they are stay in the salinity? What salinity? Is it they are stay under shade of other species, or what is their substrate? So when we understand that one, so we can transplant all the seagrass. And then of course, if we use the annual plants, might be, for example, you mentioned just now, in some months, we can see the seagrass. Why? Because they are die, then they are grow back. Their life cycle is like that. Halophila decipiens, Halophila bicri, uh, and also Halophila ovalis is a annual plants. So
Yeah.
we know that one. So that's why uh, when we transplant and halos, so that's why we mix
Yes.
with Halophila ovalis. We mix with Halophila spinulosa. We mix with Halophila major. Why? Because of that one,
Mm.
food for the dugong. Dugong cannot eat the
Mm-hmm.
halos. So that's why we mix that.
Analysis. Yes.
Yeah. So we mix.
Yes. that was, I think, that was going to be my follow-up question, but we'll come back to that because that is like one of the uncomfortable questions I was going to ask you for later. But we'll take up a few questions here. So um, here. Okay, so why did you use Anhara saccharoides
Oh,
say for a seedling and not mature transplants? Okay,
okay. Why? Because of
it's
just
a
now I
good question.
mentioned, anthers actually is dioecious. When dioecious, male and female in different plants. Okay,
Yeah.
so when we collect the matured plant, might be we collect the male only. So when we
Mm,
transplant, of course, they cannot continue their generation because we planted male or we
yeah.
planted female. So that's why when we use seedling, when we collect the fruits, in one fruit, mostly, usually, generally have 8 to 18 seeds per fruit. So in one fruit, it might be they have uh, both male and female. So we don't know. So that's why we use the seedlings. And then of course, some of the area also we can plant with the matured plant to make sure they can survive. We mix
Yep.
each other.
Yep.
Uh,
Yes. So it's yes, got it. I mean, you also increase the sample size. I mean, the bigger size you have, the better it is. Higher are the chances for you to get more stable proportions of more male and female plants both. It's
yes.
a good
And
one.
then one more thing, when we want to transplant only using a matured plant, it's very hard to pull. Very hard to collect the matured plant to pull. So because of they have very strong
Yes, with
rhizome.
extensive
As,
rhizome systems and root systems too. Yes,
yeah.
it's
So
exactly.
that's why. That's
So,
why.
other question by Aaron is how the lab culture of species is done. How do you select what kind of lab culture for what species will you do? I mean, how is that done? Just a
Okay.
very brief idea. I think Ma'am has covered that in her presentation, but
Okay. For
if you could just
example,
take 2 minutes.
okay. For examples, we study the ecology, we study the environmental.
Yeah.
We use the same situation. For example, the salinity is 28, so we try to use 28 in the laboratory. So because of that's one, and then we use a sand substrate. We not collect sand or we not collect substrate from the wild, but we use uh, sand from the near to the area because of very hard to transport the substrate
Yes.
from the Merambong whatever. So we use sand
Yeah.
from the
Yeah.
river,
It's huge.
sand from the coastal area, and then of course we mix with
Yeah. Yeah.
some fertilizer, and then we
Oh,
culture
oh,
with
yeah.
water. So my species grow well. Actually, we did this one um, since 2001 in
Yeah.
Sedang, in Faculty of Science.
Right. A quick pro tip, I think, for a newbies in restoration science would be start gardening, in your backyard.
Mm,
You'll learn the patience. I mean, this kind of work takes patience. And I
yes,
mean, the best way to do is since, getting the best kind of, whatever environmental variables you see in the natural habitats, you try to simulate them in the lab.
yes.
Next question is what physiological advantages do these lab germinated seedlings have that allow them to stabilize so much better in disturbed high sediment environments than direct seeding? I mean,
Okay.
why can't you just pull up
As we
a plant from one and a donor site and put it in another, disturbed habitat? Why do you have to go to your lab, get them germinated, and then
okay,
use it in the target restoration site? Yes.
because of in this situation, actually they are during low tide only uh, 3 to 4 months, 3 to 4 hours a day. So in 1 month, we only can sampling 3 days or 4 days. We can go to the field because of in this area actually is a subtidal shoal. Subtidal shoal, and then during
Mm-hmm.
high tide, the area is very cloudy. And then so that's why we collect the fruits, and then we bring to the laboratory to germinate first. Because of the germinating, until a size seedling, more or less 6 to 10 or 15 centimeter in length. So we can transplanting. And then if we only direct seeding, we collect seed, we have to know seagrass actually is a viviparous, and Halus ecoridis actually is a viviparous plant. So when we plant the biomes, then we plant the seed during the low tide. We planting this one, we try 100 of seed, no survival rates, because they are not
Yes.
stay in the substrate. They are stay
So
when
yes,
this one on
they
the
are
top of substrate.
positively buoyant. Yes,
Yeah, because
they'll
of the
float
oil.
away.
Yeah, so that's why
Yes.
we try. We have all this information. We think the best way is bring them to the laboratory, germinate first, and then stabilize, bring to the study area to transplant.
Uh,
Unless for
one
the Halophila ovalis or Halophila major, we not
yes, which are
bring all.
negatively buoyant. Yes,
Yeah,
yes,
we just collect 10 times 10
yeah,
and then
yeah, mm,
directly plants in the area.
yes, yes, yeah. Okay, so next question is, you mentioned record of new species in the area. What are all the factors and possible reasons which may cause
Actually,
this
it's
new
not new
species to grow?
species.
It's
It's
new
not new
record.
species, new record.
Yeah,
New record, it's not new species. Okay.
yeah.
For example, because of previously we not going to that particular area monthly, is it? So this time within 5 to 10 years, we visit the area monthly. So we start to thinking. So might be because of Halophila. Actually, Halophila species is very complex in terms of their morphological characteristics. So that's why we start to think we need to use genetic. So we study genetics. So that's why we work together with JSPS. Vietnam's, we collect together plant of seagrass from Vietnam, seagrass from Thailand, seagrass from so you we did the genetic parts, and so that's why we can prove that one actually is not only Halophila ovalis in that particular area. We discover Halophila major in 2015 together with the Thailand also, and then in 2023 we discover another species is Halophila niponica, and then because of the first original Halophila bicri first record in Peninsular Malaysia, actually in Johor in 1965. But throughout the year, we go to visit area, the seagrass of Halophila gibra, not in Johor. But in 2015, we discover that species in Sungai Pulai, Johor. And Halophila niponica, actually, we discover in 2023 by using the genetic part. Might be confusing in that particular
Mm,
time. So that's why. So might be I
yep,
can
yep.
find the new species later on. Not new species,
Yes.
yeah.
I mean, it's
New.
just a matter of, yes, exploration. You stay consistent with your efforts in mapping. Um, the more you're consistent, the more you're outside, the better are your chances of finding new records. It's not that they're new species, they're just new records. They've always been there,
Yes.
or it's just a matter of finding them at the right place at the right time.
Yes.
We have another question. What do you think the grazing pressure from megafauna like dugongs help maintaining the health for these restored meadows, or does it pose risk to the stability of new established seedlings?
Okay.
I mean, you mentioned that people have been, during gleaning of clams and whatnot, the transplanted seedlings would go to some. I mean, there are some kind of like pressure from those people too. I mean, you can't stop them from gleaning. But what about dugongs? Are they a
Okay.
problem to your restoration efforts?
For me, when the dugong come into my area, they eat my seagrass, my transplanting. I'm very happy. I'm very happy. I'm not angry. I'm happy because of uh, you must know dugong only eat the Halophila ovalis. They eat Halophila major,
Yes.
Halodiopinifolia. They eat this one, Syringodian. They not eat Enhalus. Dugong
Yeah.
don't like Enhalus. Why? I'm very sure this one because we already have studied the stomach, dugong stomach. We check the dugong stomach, the dugong
Mm.
that, and then in Johor, we get the dugong stomach, and then we study what
Yeah.
type of species the dugong eat. So that's why when we plant the Enhalus, dugong try. They are not eat the Enhalus, they eat the Halophila, the small one.
Yeah. Okay.
Uh,
So I think why this question is popping up again, and this was a question that I was in my mind too, because this was seagrass assisted and restoration effort. You're using inhalants as a stabilizer,
yes.
and dugongs. I mean, the person who's asked this question is one of my colleagues. She and Garima and Soni, there are two researchers here who have worked extensively on stranded dugongs, gut analysis, and so they have, I think, found that too. I mean, consistently dugongs avoid inhalers. I was just wondering if you're using inhalers as a stabilizer species for restoration efforts, so wouldn't there be a chance that you might change the species composition altogether? And if inhalers stabilizes better than Halophila or the dugong preferred species, so it will expand better too. It will dominate the meadows. So that could be a problem too, right?
Yeah. That's why if you want to do transplanting, we need to check what type of species there. Number one,
Mm,
we cannot simply transplant or restore the seagrass by using different species in that particular area. So we have to look at, okay, in this area we have Enhalus. In this area also we have the Samarusi. So these species we can transplant. Not, for example, we can collect Halophila, whatever other species, and then transplant in that area. So usually seagrass don't like in that particular because we
mm,
try. We try, for example, Syringodian acetifolium. We are found in Tanjung Adang. So we bring the Syringodium from the Tanjung Adang near to the area to transplant in the Merambong. They cannot survive. And also we
mm,
bring and the resilient species Halodiol pinifolia from Kelantan, and then we transplant
mm.
in the Merambong. They also not survive. I don't know why.
Why do you think that is? I mean,
Ah,
I
because of
wonder why that is.
yeah, because might be they don't like the different situations. So that's why we try to use the same species. For example, in Johor, in Merambong, we have Enhalus, Halophila, whatever. We plant the same species, but we collect the species in dense area in that same species also, that same area. Okay.
Okay. Yes, got it. Also, one last question before we move on. I mean, for everyone watching, if you have any further questions, you know where to find her. Her email ID is available on University Putra, and she's always very. I mean, she's extremely sweet. She always. I mean, I never thought that she would reply to my mails, but she did, and I'm extremely grateful for her presence. And I mean, this kind of time that she's taken to, talk about her work and her long-term work. It sure needs patience and perseverance to do this kind of work. I mean, I congratulate for this, positive restoration news to you and your, whole team. One final question from me: what's your favorite seagrass meadow? Like, just habitat and then species too.
Okay. I'm very very like Halophila bicry. Why? Okay, Halophila
Yes.
bicry because of my Halophila bicry first record type specimen actually in Malaysia. in Borneo,
Okay.
Sabah, and then I did my PhD by using Halophila bicrae. And then
Nice.
I'm discovered that Halophila bicrae, the first person in the world, is a annual and perennial. So that's why I like Halophila bicrae very much. And then in terms of, we can eat the fruits of Halus, we can eat the fruits of the Thalassia hemperchae. So that one, and also we can eat all the
Yes.
the 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 Halophila, others Halophila. But the import, I like the seagrass where I like it. For me, all areas in Malaysia, I no favorite for me. All is the same. Why? Because I'm studying all over area. Now I'm studying the mapping of the seagrass in Peninsular Malaysia. Work with the Yayasan Petronas to map seagrasses in Malaysia.
My God, it's wonderful to have you here, madam. And please stay in contact. And I
Yeah,
hope
next
that
time,
you could. Yes, ma'am.
next time we can
So,
join together.
yes, yes, I hear. Your voice is breaking up. Can you repeat that again?
Okay, next time might be we can work together. Next time we can have a discussion about how to solve the problems or how to study the biology, the
Yes,
culture of the seagrass and ecology.
yes, yes. I'd like that. I'm sure so many viewers here would love to have that conversation with you too. Thank you, ma'am, so much. With this, we are done with our second session, day 2 of World Seagrass Day. Join us tomorrow again for another talk, this time with implications to dugong conservation. Um, across Southeast Asia, we know whether you're working for dugongs, whether like me, you're working on dugong herbivory, or foraging behaviors, whether you are exclusively working on dugong behaviors, you cannot, work without addressing questions of habitat restoration. If a habitat is gone, so will your dugong. Site fertility changes. So I'm sure with what we've learned today and what ma'am has spoken at length, we'll also sit down, introspect, and reflect with, in terms of dugong conservation and movement ecology and foraging ecology. Thank you, ma'am. Thank you so much. It was quite beneficial to all of us.
Thank you. That's all.